forced Darwin finish his book. But before that, in the August of 1858, both Darwin and Wallace published a paper together on the tendency of species to form varieties, on the perpetuation of varieties and species by natural means of selection. Yeah. This is 1 year before Darwin published The Origin of Species. And what are the principles on their theory? The principles go back that there is phenotypic variation. Yeah? So individuals of a population are different. Yeah? There are limited resources available in the environment, and organisms basically compete for those limited resources, which results also, and then there's survival of the fittest, which is explained by natural selection. They didn't know anything about genetics, and they focused on quantitative traits, so morphology and so on. By having these large collections and by observing like individuals of a single species and thinking, okay, I have this beetle, I have 100 specimens of one beetle, are these hundreds which all have slight differences? Are they all individual species? As the historic view on organisms was, everything was just originated and existed? Or is this one species with a lot of variation? And that's what they realized. And so today we will talk about how this variation is coming into existence. So the evolutionary theory of Darwin is the following: in a nutshell, organisms keep on evolving. There's a non-constancy of species. This is the basic dogma. Yeah. Nothing is fixed, everything is changing all the time. All organisms descend from a common ancestor. And there is a multiplication of species over time. So Darwin's theory can explain biodiversity. And evolution is not a sudden change, it's a gradual change, and it takes place all the time in every generation, as we will see today. Yeah. So no so-called saltations. This is the sudden appearance of a new organism or a new morphology. And evolution is based on phenotypic variation, the competition for limited resources, and a process which we call natural selection. And natural selection is essentially the selection by the environment, which includes, again, biotic and abiotic entities, as compared to artificial selection. So Darwin was working closely, besides working with his collection and with naturalists, he worked a lot with animal breeders. Yeah? And if you have a dog, you know that your dog has specific traits. No, there are so many breeds of dogs. They are still the same species, but we can breed them. We select for specific characteristics or traits, whether it's the size, the color of the fur, whatever. Yeah, this is artificial selection, so we can see that phenotypes can be very different, even within one species. So the main issue is, however, that Darwin never figured out what the source of the variation was, which produced different individuals. Yeah, and he wasn't able to ever figure that out. We will hear this today. Yeah. When Darwin and Wallace had written their work up and were revolutionizing the thinking, soon after, due to the impact of Gregor Mendel's genetic work, the Mendelians were a group of biologists who basically throw over Darwin's what they called old-fashioned speculation on natural selection, and simply said that no, all the species originate from mutations alone. Mutations are the thing which drive everything. Yeah. So this was around 1900, 1910, when this perspective was like very dominant in biology. And it took another 20 years or so before our, I would say, modern perspective on evolution was developed in the so-called great synthesis or the modern synthesis, or sometimes also called the evolutionary synthesis. Yeah. This was a time of very active research and publication and debate over about 20 years. And those people who are shown here, Robert Fisher, Jason Huxley, Thomas Dobzhansky, Ernst Mayr, Stebbings, and Simpson, they were all basically bringing in some of their views from population genetics to systematics, from zoology, from paleontology, from botany. And they synthesized their views. In particular, they synthesized the ideas of Mendel on genetics and the ideas of Darwin on natural selection. Yeah. And so basically, you can summarize their ideas that mutations create the variation which selection acts upon. Yeah. So it's neither nor mutations or natural selections, it's a combination of both. Okay, so now look into some molecular foundations. Let's remember things we've learned in high school, and you might have heard these things already in your introductory semesters if you study biology. We talk about how we go from DNA to a phenotype. So those things, those molecular and cellular processes that Darwin didn't know. Yeah, and I do this because I want to make sure that also the students of geoscience know when we talk about what we mean by a gene, by very principal concepts. Yeah. So we start with where we find the genetic information. It's condensed in what we call chromosomes. Yeah. These chromosomes are located in the nucleus of cells in every eukaryote. So every organism where a cell has a nucleus contains chromosomes. They're typically only visible when cells divide in their kind of like cross form. And when a cell division is occurring, we have a process called mitosis, and a chromosome then consists of two arms, two chromatids, which have a single strand of identical DNA. Yeah. So if you would take apart all the chromosomes and stretch out the DNA, it would be a 2-meter long molecule. So that's why it is really condensed in these chromosomes. Yeah. So here, just for those who don't remember this, you know, when we divide a cell, before we do this, we replicate, we double the amount of DNA in a cell, and then we form these cross-shaped chromosomes. Yeah. We are diploid organisms. That means we have a set of genomes from our mother and our father. Yeah. And in order to give them to every daughter cell, we need to double them and then divide them. Yeah. So every cell in our body has the same gene, contains the same genome, the same number of genes, in order to know what the cell has to do. Yeah. And think about your hair grows, your skin renews, your organs renew. So your body is constantly working. So every cell needs basically genetic instructions of what to do. Yeah. So humans have 23 pairs of chromosomes, and we have, as I said, a diploid set. That means we inherit one set of chromosomes from the mother and one from the father. So the individual set of the what we call the maternal for mother and the paternal for father chromosomes are haploid. Yeah. But they are what we call homologous. They carry the same set of genes at the same positions. Yeah. So this is important to realize. So let's look into the chromosomes then. That's where we find the DNA. And the DNA carries genetic instructions to make, basically cells and to make living organisms. DNA is a macromolecule composed of two chains which run in opposite directions. They contain nucleotides and they form this, what we call a double helix. Yeah, you see this in this animation here. And what is a nucleotide? That basically is a part of this molecule which consists of a nucleobase, sugar, deoxyribose, and one phosphate group. And remember, we have these four nucleobases: adenine, cytosine, guanine, and thymine. So every organism, whether it's a bacterium, archaea, mammals, birds, plants, they have the same, these same bases and these same molecules. DNA, besides proteins, lipids, and carbohydrates, are the four, or are these major molecules which make up organisms. They're essential. You will not find DNA isolated on Moon or on another planet. These organisms, these molecules, you will find only in organisms. They are not synthesizing themselves in the environment. So here a view, we have DNA, we have RNA. DNA is a double strand. And we have to remember, we have these bases which are complementary. And we mean, what does that mean? Complementary molecules are molecules which can form bonds. And you know, in the center of this, try to use the mouse here, in the center of this DNA molecule, we have hydrogen bonds, which can only form between these complementary base pairs, which is cytosine or guanine or adenine and thymine. The others don't work together. So this is important to realize. So when every cell divides, or before every cell divides, we have a process which we call DNA replication. This is it's the basis for biological inheritance. That means, and it's very cool to have a DNA as a double strand because it opens and it can be read off by an enzyme which we call DNA polymerase, which produces then a daughter strand of exactly these complementary bases. And so we can divide the DNA molecules into two. Half of the parent DNA goes into one cell with then newly synthesized opposite strand, and the other half with a newly synthesized opposite strand goes into the other daughter cell, or remains in that cell. So this is a process which occurs before every cell division, whether it's the division of somatic or germ cells. So, and it's necessary to maintain the genome in an organism or in a multicellular organism. So DNA replication is a process by just replicating, doubling the DNA. And the biologists may know that there's a lot of biochemistry involved in here, but there are processes which ensure that the right, when the new DNA is synthesized, that the right nucleobases are always used. So there's normally a very low margin of error. DNA is also found in organelles of the cell, which we call mitochondria. These are organelles which produce energy in the cells. And mitochondria, interestingly enough, have their own genome. So they carry their own DNA, which we call mitochondrial DNA. That goes back to some idea that mitochondria were once independent organisms, and they formed a symbiosis. So their origin is one theory. The endosymbionts theory proposes that mitochondria were, I think, bacteria originally. So in any case, they have also DNA, and we can sequence this DNA. And in humans, this DNA was relatively short compared to the nuclear DNA, the DNA occurring in chromosomes in the nucleus. In humans, it encodes for 13 proteins, for example. Importantly is that the mitochondrial DNA is only inherited from the mother. So this is important because then we can use this mitochondrial DNA to look at the maternal lineage, which is now often used in phylogenetic studies. So mitochondrial DNA evolves comparatively fast compared to other nuclear DNA markers and has been widely used now in all kinds of phylogenetic studies, which we will learn and read about in this class later. So many phylogenetic trees we will come across will use mitochondrial DNA. One of the results of sequencing mitochondrial DNA is that biologists predict that all living human beings, so all of us in this class and everybody else alive on this planet, goes back to one mother, which is called the mitochondrial Eve. So we all descend from one single female human being, which can be predicted by looking at the difference of the mitochondrial DNA. And this mitochondrial Eve is hypothesized to have lived in Africa 150 to 200,000 or 300,000 years ago, which fits well with the fossil record of Homo sapiens. And it also rejects all the ideas about racism and everything, because we all originate in Africa from an African person. So we know now, hopefully everybody is on the same page, and please interrupt me or raise a question if you can't follow what we mean by DNA. So maybe I should mention in plants, DNA is also found in chloroplasts, which sometimes botanists use. But let's think about what we mean by a gene. So a gene is essentially a sequence of DNA or RNA that encodes for a molecule, and these molecules are in most cases proteins. So these genes are located on fixed positions on the chromosomes or on the DNA sequence, which we call a locus, on plural loci. So we can map them and build up, by doing this, a gene map. This has been done for Homo sapiens and for many other organisms. As I mentioned before, we are diploid organisms. So we have a set of genes from mother and father. That means every gene is present in every of our cells in 2 copies. So we have 2 copies of each gene. And I should mention maybe that all sexually reproducing organisms are mostly diploid. There's polyploidy, so there's some plants which have more sets of chromosomes than just 2, but let's just stick to that. Because we have 2 copies of each genes, they can be different. So each gene can be different in can be present in a slightly different way, slightly different sequence of DNAs, and then we call this an allele. So alleles can encode for slightly different versions of a protein. They don't have to, but they can. And this may cause, not must cause, but may cause different phenotypical traits. So having alleles is very important in what we will see, the origin of phenotypic diversity. Genes also include other aspects of DNA. One are exons. Exons are the part of the gene which will actually be encoded into a protein. So it's the expressed region of a DNA. And you must realize that these exons, which are essentially those parts of the DNA strand or the genome, which are encoded in proteins, only make up about 1% of the human genome. Genes also encode what we call introns. So these are parts of a gene, of a DNA sequence, which is removed during transcription and not used for coding proteins. So 24% of the genetic genome is made up of introns, so of material which is not used. And in addition to these introns, there are also intergenetic regions on the genome, which we call non-coding DNA. So there's a lot of DNA which is not used as genes to make proteins. Yeah? Biologists are working actively on figuring out what this DNA is doing. Sometimes it has a regulatory function, but so far we realize, or researchers realize, that maybe only 10 to 15% of the human genome has a biological function. Yeah. And on Monday, we heard that the term biodiversity, or the concept biodiversity, encompasses genetic diversity. And we also heard that there's a difficulty sometimes to relating genetic diversity to the biodiversity we see, for instance, expressed in species. And one of the reasons is because of this non-coding DNA. There's a lot of DNA which we cannot understand because it's not used to really constitute organisms, cells, etc., and thereby the phenotypes. Yeah. So how do we actually make then proteins and which will become our organisms? This is a process called gene expression. Yeah. So this is the fundamental process of expressing the genotype into the phenotype. Yeah. And this process involves two steps: transcriptions and translation. And you probably remember this from school. If you're not studying biology, I will quickly go through these things. So transcription basically is a process which starts in the nucleus. The DNA, which is like stuffed together in these chromosomes, is too long to leave the nucleus, so small portions of it are being read off, which are RNA molecules. Yeah. So we split the DNA strand, and then from one strand, which is the template strand, we read off the complementary nucleobases and form a strand of RNA, which is exactly what the opposite strand of DNA is, so the coding strand. Yeah. And then this messenger RNA is going out of the nucleus. Yeah, it carries the information of the DNA. So then we have a process of which is called translation. Yeah. The second process of gene expression, and it translates essentially the nucleic acid language into what is called the amino acid language. So no new molecules come into play. We have these DNA and RNA molecules, and now we build amino acid sequences which make up proteins. Yeah. So what are these proteins? So they are made of peptides, of organic compounds which consist of amino acids and linked by these peptide bonds, as you see here. And if such a peptide is being built and you build a chain over 100 or so amino acids, you build a protein. Yeah. So proteins are essentially just long chains of amino acids. And we have only 20 standard amino acids available to choose from to build all the proteins which we need. Yeah. And these proteins are then involved in most biological processes. So the functioning of cells, of organisms, requires these proteins. Yeah. So how does it go? How do we go from the DNA sequence of nucleobases to an amino acid sequence? So in the nucleus, we transfer DNA information, the genetic code, to messenger RNA. This code tells the messenger RNA which amino acids to use to build a protein by basically a sequence of 3 nucleobases, which we call a codon. Yeah. So you see here a strand of RNA, and you see the respective nucleobases in sequence. And 3 of them, which you can call a base triplet, form so-called codon. This is the genetic code, essentially, written in sequences of 3 letters. Yeah. The translation, this process of translating this DNA sequence into the sequence of amino acids takes place in what we call ribosomes. Those are outside the nucleus, and it requires another type of RNA, so-called transfer RNA. This transfer RNA molecule is shown here in the upper right, and it carries 1 amino acid. And this transfer RNA has what we call an anticodon, so basically the complementary sequence of the messenger RNA sequence, but a codon. Yeah. So 3. And so it comes with this amino acid, goes to the ribosome, and then there a chain of peptides is being synthesized as a result. So looking at some amino acids, yeah. So what we have, what we call proteinogenic amino acids. These guys can build proteins. Yeah. There are 21 in eukaryotes, and 20 of them can be encoded with standard genetic code. Yeah. We as humans can synthesize only 12 of them in our bodies. The other 9 we have to intake with our diet. That's the reason why people who want to build muscles use protein shakes, which contain these, what we call essential amino acids. Yeah. So we can then plot the letters of a codon, so of this base triplet from the DNA sequence in this matrix. And in the center, we plot then the resulting amino acids. You see in this matrix that there are several synonyms of codons available to form an amino acid. Yeah. So the 3rd letter of the codon can vary, and you result in the same amino acid. This is what we call a degenerate code. Yeah. So you can only read the code in one direction. If you have a sequence of nucleobases, you know, of 3 of them, you know which amino acid to build. If you have, let's say, alanine, yeah, this amino acid, you cannot go back and say from which codon it was synthesized. This is what we mean by a degenerate code. Yeah. So because there are 4 possibilities to form this amino acid. So all forms of life use the same genetic code. This is one of the pieces of evidence we have for common descent. So this is so fundamental to life. Yeah? And a gene usually consists of many codons. So there are very few examples where maybe only 1 or 2 codons actually can function as a gene. Yeah? And each protein contains a different relative proportion of the 20 standard amino acids. So from 20 amino acids, we build all the proteins we need to have life. And the codon is, again, is a set of 3 nucleobases out of 4 possibilities. So what does it mean? That gives us 64 possible codons, yeah, 4 to the 3, to translate into 20 amino acids and 3 stop codons, which we need to build proteins. So if a codon would only consist of 2 nucleobases, we could not build these 20 amino acids. So having 3 is essential. But as you see in this figure, this result in codons which are essentially, you would say, redundant, because they can form the same amino acid. So this is another view. This is a so-called DNA codon table. If you studied biology, you've seen these things, and you probably had to learn every sequence to form an amino acid. So this is all not new to you. You go from the inner to the outer portion of that ring, and then you can read off your specific amino acid. So in a nutshell, this is how we go from the genotype to the phenotype. Obviously, there are several other aspects and pathways related to then going from a protein to the actual appearance of an organism. But basically, the DNA provides us a code with which we can select for a limited number of amino acids. And from these 20 amino acids, we can build all the proteins we need to then develop the organisms with all the diversity in terms of their appearance, behavior, and so on. And so this is the basics for understanding how the code written in DNA is being expressed. So a few words, just as a side note here, on the role of amino acids in phylogenetics. Most people think about, we've heard about mitochondrial DNA can be used to reconstruct phylogenies, but also amino acids are used. So phylogenetics, for those who have no idea what this is a field of evolutionary biology which studies the history and relationship of individuals or group of organisms, species, populations, etc. And besides DNA sequences, we can also use amino acid sequences of specific proteins to decipher the evolutionary history of organisms. So we don't just need DNA sequence machines, we can also sequence amino acid sequences, or sequence proteins essentially. And when we compare those sequences using widely distributed proteins, we can reconstruct phylogenetics or phylogenies. And why can we do this? Because similar to mutations, which we soon talk about, which change a DNA sequence, there's what is called an amino acid substitution. So one amino acid is being substituted once in a while. And this occurs at constant and typical substitution rates, in particular proteins. And if you know the substitution rates, you can actually, and you compare the sequence of amino acids for a particular protein from different organisms, you can reconstruct the history of their relationships. This is shown in this example. This is the protein cytochrome C. And this protein has a substitution rate of 1 substitution in 100 amino acids over 20 million years. So you see, it's very slow. And the protein is made up of 104 amino acids. Plants, fungi, and animals which have been sequenced for these amino acids show that 19 of them are still in common. That shows us that there are this interrelationship between all these organisms. If you look in detail between, let's say, the first 4 groups, so you look at, if you look carefully at the sequence for humans, chimpanzees, spider, monkeys, and macaque, so all this letter denotes 1 amino acid. You will see there's no difference between humans, chimpanzees, and macaques. There's a difference with spider monkeys. This is in line with the timing it takes to substitute one of these amino acids in this protein, because humans and chimpanzees diverged only, I don't know, 10 million years ago or 8 million years ago or so. Macaques, probably not much earlier. In spider monkeys, monkeys in South America. So they, I think, South American monkeys, New World monkeys, invaded South America about 30 million years ago. So they should show some signs of divergence. So you can use these amino acid sequences also to develop phylogenies. Yeah. This just as a side note, it's not essential to our understanding. But I thought because we mentioned these amino acids, we can say a few words about them.
You were coming from Africa?
Originally, yeah.
Yeah. How did you cross?
Rafting. Okay, let's take a break of like 2 minutes to digest all this. And this is also an opportunity to ask questions, because the next or the last part of today is talking about mutations and reproduction. Let's talk about a few other principles which are essential to understand phenotypic variation. Yeah, and one of those processes we call mutations, so which is spontaneous change of the genotype, a sudden change of a gene. Yeah. So, and the mutation is not reversible, so it's a permanent change in the genome. So mutations can only affect an organism's phenotype if they occur on the protein coding sequence of a gene. That should be understandable also now, right? Because we heard there's a lot of DNA that does not encode for proteins. And there can be errors in the replication of DNA during cell division or the formation of germ cells or gametes, which then result in so-called gene mutations. Yeah. And a gene mutation is essentially the replacement of a single base pair by a different one. Yeah, I soon have a few illustrations of that. And the error of these, the rates at which these errors essentially occur is very low. Yeah, so 1 in every 10 to million bases. So it's not a common phenomenon. There are also chromosomal mutations which will change larger portions of the genotype. Yeah. So there are different types, and we don't need to learn them in this class. Obviously not. You just must understand that not every mutation has an effect. Yeah. It really depends whether the mutation affects protein coding region or not. And even if it affects one, it does not mean that it will change the protein which is being encoded. Because you've seen we have a lot of redundant codons. So that means different codons which will produce the same protein. However, proteins can be truncated during their synthesis when we insert a stop codon, or the whole sequence can be changed. Yeah. So, which is, if we have a substitution of one base, we have a so-called non-synonymous substitution, or sometimes called missense substitution. Nonsense substitutions or mutations result in a stop codon, and these are point mutations occurring in one single location. And then we can also have silent substitutions, so a mutation, as I said, which does not change the amino acid sequence. Yeah. So here, this is shown here. We have a DNA coding strand with a sequence of codons and the respective sequence of amino acids. If we have a silent mutation, we change one letter, one base, which still results in the same amino acid. So we are not changing the protein, has no has no effect on the organism. If the substitution is changing the amino acid, we may encode for a new protein. So this can have an effect. We can also produce a stop codon, which are a few codons which result in the termination of building a protein. And we can also have a frame shift, that is the deletion of one base, and which then also results in a different amino acid sequence, probably different, or some error of the protein. So here you have this for like kids to read how you can understand this. I'm not going to read this aloud. Just you have this on the slide, and you can see what this means in very simple terms. Yeah, for those who have no biological strong background, this is very easy to understand it. But essentially, mutations that induce changes in the phenotype can either be favored or discriminated against by natural selection. Yeah, Darwin had no ideas about these mutations. We know they occur, and we know that they have three types of significance. They can be beneficial, they can be neutral, or deleterious. Yeah. So beneficial are all those which are favored by natural selection. Yeah. So think about your bird, a male bird, and all of a sudden you change your plumage to a very brilliant red color among otherwise gray males from one mutation. You may attract a lot of females, and you have a lot of offspring. Yeah. So that could be beneficial. It could also be deleterious, because the red color may attract more predators, and you're lost within one generation. Yeah, so natural selection is then something we talk about next week to understand what that means. Yeah. So neutral mutations are those which affect non-coding DNA. You heard we have a lot of non-coding DNA. So these mutations really do not affect organisms. They do not affect the fitness, the chance to survive the environment. And if they are deleterious, they're either recessive, they can survive in a population, or if they're dominant, then they're lethal. They will eliminate themselves because the organism may die. Yeah. So without mutations, all forms of life on this planet would be genetically identical. Yeah. So this is so important to understand why there are different forms of life, because otherwise they would carry the same genetic code. And every evolutionary so-called innovation originates from a single mutant allele. Yeah, so think about all the diversity in form and color and life forms and so on goes back to these mutations. So that's why you can also envision that it takes a tremendous long amount of time to build diversity, because it's a slow process. On the other hand, we should not overstate the relevance of mutations. For instance, in a population, the frequency of genes is rather influenced by natural selection and genetic drift, and not just by mutation. So we hear about this next week. And then also, we should realize that most phenotypic variation, so the variation of individuals of a population, is produced mostly by recombination and not by mutations. So what do we mean by recombination? This is the last part for today. So recombination is part of sexual reproduction. So organisms which perform asexual reproduction essentially form offspring which has the genetically identical makeup. They are what we call clones or copies. Yeah. This is the most common type of reproduction in single-cell organisms such as bacteria. Yeah. This has a number of advantages. It allows for rapid population growth because everybody can reproduce, not just female organisms. So you can have twice as many offspring. Yeah, so bacteria grow so fast if the environment provides the right conditions, much faster than other organisms. Also because these organisms don't have to spend or allocate resources for reproduction, which all other organisms do, either to attract a mate or to carry or to care for the offspring. So there's a lot of costs involved. For those among you, like Guillaume, who have children, know that it costs a lot of energy to bring your offspring up. Yeah. So this problem is not present in asexual organisms. But they have the problem with that all the genetic diversity only originates from mutations, which, as we have heard, are not so common. So they evolve even slowlier. So what do we mean by recombination? Yeah. So this is a fundamental aspect of the formation of germ cells. So egg and sperm cells, for instance, in mammals. Yeah. So recombination is present in all sexually reproducing organisms. And what do we mean by this? It is the rearrangement of genetic material, which is inherited by mother and father. It is in particular the exchange of alleles from those maternal and paternal chromosomes in the cells during the process of meiosis. Yeah? So meiosis is the formation of egg or sperm cells, and this meiosis, or before these sperm cells and egg cells are being produced, they are preceded by 2 cell divisions. Yeah? So during the first division, the chromosome inherited from the mother and from the father attach to each other and break at several places, and they exchange part of their DNA sequences. You heard because because the genes are located on specific fixed positions on these loci, there will be an exchange of genes, and there will be not like a mess. This process of exchange we call crossing over. And then we have a first cell division of these chromosomes to 2 daughter cells. This is shown here. Yeah. So before we do the meiosis, we double the DNA. We have DNA replication. Then we have the chromosomes present in these double forms. The arms, the corresponding chromosome arms, they overlap and they exchange part of their code. It's a full exchange. Yeah. And then we have a cell division, and we have 2 diploid daughter cells which contain a new setup of genotypes. Yeah. Then we have a second cell division, which we call a reduction division, where we form 4 haploid gametes, 4 haploid germ cells. So by doing this, we have a result. We have gametes, so cells which each carry a new and different combination of parental genes. Yeah? So each germ cells which is produced in each sexually reproducing organism contains a different sequence of DNA. Yeah? So this is so relevant. So you see it here, the whole process. After the second division, we have 4 haploid daughter cells. So whether this is an egg cell or a sperm cell doesn't matter. Each of them has a unique genotype. So over time, this process, meiosis, dissolves individual genotypes. Yeah. So no genotype, no sequence is surviving for long over generations because of this rearrangement, this recombination. Yeah. So for those, let's say, maybe not the biologists, but assume there is no recombination taking place in a female and male organism. Yeah. So one generation just doesn't do this, what I've just explained. And then these two individuals come together and they mate and produce offspring, a large number. What would be the genome of the offspring? Any ideas? Just throw something out. It's completely hypothetical. But I want you to think about what I just tried to explain.
Well, I guess the genome of the offspring would be identical to that of the parents. If the female and the male organism don't recombinate their genetic material within the offspring, then the offspring would be identical to the parents. There would be like clones.
So let's go back to the previous slide. Yeah? Just, I know you're probably tired and have heard so much theory today, but mother or father inherit their genotypes from their parents, right? So what you see in the very top here, let's say this one here is a cell of the mother, and it has a chromosome from her mother and her father. Yeah, so they don't overlap, they don't recombine. So the 4 haploid daughter cells will be basically representing half of the genotype of, or basically genotype of the mother, of the grandmother, the grandmother, the grandfather, and the grandfather. And if they combine, you have a mixture then of the grandparents. Yeah. So because of this process that we have recombination, we never mix these things up. We never produce these copies or so. So each generation is different. So when there's fertilization, these 2 haploid gametes form a diploid zygote, and this diploidy is basically restored. And for those who haven't studied biology, there's no fusion of the parental chromosomes, or no fusion of the DNA sequences, so they remain discrete. Yeah, all the variation which comes into existence, it goes back to this recombination prior to the meiosis. So the advantage of this mode of reproduction resulted that most organisms, in terms of diversity, species diversity, for instance, most organisms perform sexual reproduction. And within the life cycle of these higher organisms, this process is assumed to be one of the most important events as a result. So the significance of recombination in higher organisms is as follows. It leads to new gene and trait combinations. All individuals in a sexually reproducing population are genetically unique. Yeah? So recombination just removes similarity, essentially. And together with mutations, reproduce genetic variation in a population. Yeah. This is the variation which Darwin and his contemporaries had no idea how it originates. Yeah. It is important to understand that sexual reproduction results in many more new phenotypes in a population than would result from mutations alone. Yeah. So think about your siblings, if you have them, or think about relatives, everybody looks different. And this goes back not necessarily to mutations, but to recombination. Yeah. So the genetic variation, which results from both mutations and recombination, is the very foundation for adaptation of organisms to changing environmental condition in a process which is called microevolution. And so the success of this way of reproducing is evident in the absence of a return of higher organisms to asexual reproduction. Yeah. So it's really that natural selection is actually favoring it, because you produce all kinds of gene and trait combinations, all kinds of phenotypic variation, and in that way, there's a high chance that certain individuals are better adapted to changes, either in the environment or to disease, etc., and have a chance to survive. So the person who figured these things out in terms of the relevance of sexual reproduction was Gregor Mendel. He found that different alleles of genes account for all the variation in inherited characteristics. So we get these two alleles from mother and father, but we also have then usually one trait expressed. And this is why either because the alleles are the same, which is called homozygosity, or they're different, heterozygosity. And if they're different, then the alleles can be dominant or recessive. Yeah, so either or. So again, some key concepts here to understand what we mean by homozygosity and heterozygosity. So alleles are just a different form of a gene. If both gene or forms inherited by the parents are the same, we have a homozygous trait. Yeah? If they're different, we call it heterozygous. Yeah? And the presence of heterozygous traits is also called polymorphism. And we can count what people are doing, molecular biologists and so on, geneticists. They can call they can quantify the frequencies of alleles. For instance, within a specific locus for one gene in a population. And then you can say something about how fit a species is, for instance, or population. So Mendel performed his famous experiments with peas, yeah, and you denote an allele which is dominant by a capital letter and one which is recessive with a small letter. And so we have this way of denoting them. There can also be a situation where you have codominance, where both alleles are being expressed. Yeah? So the blood types of humans are determined by alleles, and the A and B allele are codominant. So that's why we have an A-B group as well. Yeah? Or we have other things. Just as an example that this is actually relevant, it's not just some theory of people who breed plants or animals. This is a study came out a few years ago on the genomes of gorillas. So the entire genome from mountain gorillas and other gorilla subspecies had been sequenced. And they found for mountain gorillas, where only around 1,000 individuals are left in the environment, that they're homozygous in 34% of their sequence. Yeah? That means their population is very small and there's a high level of interbreeding. So if you made individuals mate with a closely related relative, the chance to have a similar gene sequence are high, so the chance of having the same allele is high. Mendel performed his experiments, and what is relevant, what he found out to when he did these experiments of breeding these plants, is that you have these dominant alleles which will all be shown in the first generation of offspring, which is here shown as F1. Yeah. But if you then cross individuals of this first generation and you produce sufficient number of second generation plants, because they still carry these recessive alleles in their genomes, there are two things. First, these recessive phenotypes become again expressed in individuals, but you also form new phenotypes, round and green, wrinkled and yellow. Yeah. So this is why evolutionary biology concerns populations and is concerned with like longer timescales. We are not thinking about individuals, we always look at the populations. So over time, things can rapidly change, or they are not expressed in one generation and can be then expressed in the next one. Yeah. So in summary, sexual reproduction involves the recombination of genetic material. It results in a continuous change in the genetic composition of every population. And if this combines favorable genes, there's a high chance for fast adaptation to environmental change. And we will see in this class that various organisms or various species are either, and often actually today, in a situation where they are declining. And one of the reasons is because their populations are artificially or for other reasons being reduced. So when you think about sexual reproduction, it is important to keep in mind that this is a good process. It helps to produce new phenotypes, but it also depends on the number of offspring and the size of a population. And these are topics we are going to talk about next Monday. Yeah. So I think I skipped this slide and just want to say that the advantages of sexual reproduction are higher adaptability, higher phenotypic variation, a greater chance for mutation, and a greater chance for survivability under environmental change. Yeah.
Can I ask something? Do you hear me? So yeah, it's a basic question. I think I know the answer, but maybe for the geologist, it could be like interesting to know. In case of these gorillas that they have 30% of homozygosis, what will that represent in terms of parasitism, adaptation, diseases? I don't know, maybe you can say something about it.
Yeah, so it shows two things. First of all, it shows that they underwent a population decline over a very long time already, not just the last 100 years or so, as a result of human impact on the habitat. So the habitats of these mountain gorillas is severely reduced, but they undergo a population reduction over thousands of years. But it's interesting, they seem to be adapted to these high altitude environments. So apparently then the alleles responsible for part of these adaptations are favored. Yeah. But as a consequence of homozygosity, it can result that if there's a certain disease breaking out, that they could be at a very severe disadvantage. Yeah, because they perhaps lack the genes required to be resistant to some new types of disease. Yeah. So it is not necessarily how to say, very negative, as I understand the paper. Yeah. So the authors claimed the homozygosity could be a sign, obviously also is a very narrow adaptation to the mountain habitat they live in. Yeah. And they found, for instance, that if I remember correctly, there was one gene which is related to high altitudes and I don't know, something that related to respiration or so, and levels of oxygen, that this is so similar among the individuals. Yeah, is this probably a sign of being, forced into this high altitude environment? So it's not necessarily all negative. Yeah, but obviously if there's a new factor of change in the population, or a new impact climatically or disease-wise or so, then obviously there could be a severe or dramatic impact on the entire population. Yeah. So maybe I post a paper. We'll use that paper a couple of times. And you can read this if you're interested.
Yeah, cool. Thank you. Yeah, that was more or less like what I was thinking.
Okay. But what are your ideas, Jose?
No, it was similar. Like if there's like a vector disease that, yeah, suddenly came from outside, there's a higher risk that the population get hit very hard, and it could lead to extinction in a really severe case, or yeah, like you said, also maybe in the other way. I didn't know that. That was actually interesting what you said about this allele of the oxygen that could be like prompt by the homozygosis, and yeah, I didn't. That's interesting to hear about it. But yeah, it was more about diseases and adaptation that I was thinking.
Yeah, so I mean, a recessive allele doesn't mean it's not one which has an advantage. Yeah? And because the environment is also constantly changing, if we, go out of our human perspective of time, then different alleles, a different combination of alleles are favorable under certain conditions and of disadvantage on others. So in any case, I like to summarize, and then we have a couple of minutes maybe to clarify some other aspects. So for today, again, very basic introduction into some of these principles. We should know that the gene is the functional unit of heredity, and the genes determine the traits of an organism, which we call the phenotype in all its appearance and so on. Every cell carries a whole set of genes, one from the mother and one from the father in diploid organisms. The genetic code translates information into proteins via the codon. Yeah. And during cell division, every gene is doubled. We have the process of replication, and each copy is given to the daughter cell. So every cell of an organism contains the same set of genes. Yeah. And if we have sexually reproducing organisms, we can produce new combinations of genes through the process of recombination prior to the formation of gametes. Yeah. And changes in the genetic code can be introduced by mutations. And mutation and recombination in combination are the precondition for evolution to occur. Yeah. So these very basic, and maybe it's a very dry topic, I apologize for this, but it's just fundamental. This is the foundation to understand then aspects of how the environment can change populations, how random effects can change populations, and ultimately how speciation can occur. Yeah. So without having a tool essentially to produce variation in the genomes and therefore variations in the organisms or in individuals, we wouldn't be able to have diversity. Yeah. We would have setups of molecules which just probably replicate themselves and never be different. Okay. I'm at the end for today with this lecture.